morning, everyone. My name is Boon Hua, marketing specialist from Scripps, and we are the world's leading biotech company with gene synthesis, protein, antibody, and preclinical drug development service capabilities. In today's webinar, we are going to discuss the customized antibody generations for the therapeutics and diagnostics research applications. So let me introduce our speaker today, Dr. Hao-Wing Leong. She's the field application scientist of Scripps, who specialize in protein expressions and antibody production services. Dr. Leong graduated from the National University of Singapore in 2011, majoring in pharmacology and neuroscience, and worked as a research fellow in Duke and US Medical School thereafter. Dr. Leong has more than 10 years of research experience in molecular biology techniques and extensive knowledge in protein structure modeling, protein expressions, and its purifications. This webinar will take around 40 minutes and it will be a recorded session. You can type in your questions in the Q&A box and we will answer them after the presentations. If we didn't get to answer your questions during the Q&A sessions, we will be sure to take down and email back to you. So without further ado, let's welcome Dr. Leong. Over to you.
Okay, thank you, Boon Hua. Thank you for the kind introduction. And good morning, and welcome to the webinar. I'm Hao-Wing. I'm the field application scientist for Scripps protein expression and antibody production portfolio. For today's presentation, I'll be providing you with an overview of Scripps customized antibody generation for therapeutics and diagnostics research applications. We take pride in being the number one gene synthesis provider in the world. From the natural extension of our gene expertise, we are well positioned to become a custom antibody service provider and are honored to be voted and awarded as the most popular custom antibody supplier by CyTab. We have extensive experience in customized polyclonal, monoclonal, and anti-idiotypic antibody modification, and also antibody drug discovery. We have about 3,000 employees globally, and 30% of them holds a postgraduate level degree. Scripps have been cited in over 30,000 peer-reviewed journal articles. And we at Scripps offers a comprehensive service portfolio to scientists in more than 160 countries, ranging from gene synthesis services to peptide, protein, and antibody services, and a full array of COVID-19 solutions for vaccine development and beyond. So for today's presentation, I will give an introduction to the Scripps and antibody platforms, some of the case studies that we have for custom antibody cases that we had in research, diagnostic, and also therapeutic applications. And lastly, what are the other antibody-related specialized services that we have in Scripps? We are more than a professional service provider, and our highly skilled scientists may help shape your research program to the next level. So first, let's start the presentation with Scripps antibodies platform introduction, many of which are state-of-the-art capabilities that are proprietary and unique to Scripps. A brief look at the history of the antibodies first indicates that we have come a long way since the Nobel Prize awarded to Emil von Behring for their landmark work on showing the serum transfer from immunized animals and how it could cure diphtheria. And since then, many research have been performed to understand antibody and to optimize the process for its generation. And the work by Georges Kohler and Cesar Milstein in 1975 with their work on hybridoma technology actually signifies the start of the modern era of antibody research and discovery. So in Scripps, we are also proud to be utilizing some of the platforms, and we are always in search of the development. We are proud to be to provide the one-stop solution for all custom antibody needs. We capitalize on the animal's natural immune system to generate quality antibody leads with high specificity and affinity thanks to the intrinsic affinity maturation abilities for polyclonal or monoclonal antibody generation. And how do we do that? So GenScript has developed a series of proprietary antibody technologies to help ensure the success in generating high specificity and high affinity antibody that maximize your research potential. Our ultimate antigen design program is using an advanced antigen design algorithm to optimize the likelihood of surface exposure and antigenicity. ImmunoPlus technology makes use of key immunomodulators to circumvent self-tolerance mechanisms and to allow B cells to generate high-affinity surrogate antibodies. And we also have epitope binning, which is an independent antibody validation in which monoclonal antibodies are tested against one another to determine which antibodies compete for the same antigen epitope. GenScript also has a proprietary adjuvant system to quickly elicit strong and durable immune response to generate high specific antibodies. Last but not least, the express immunization and high efficiency electrofusion allow antibody generation with speedy turnaround and of large quantity and also large diversity. GenScript has also established state-of-the-art animal facilities with ALAC International and OLAW certification. Our facility can provide a broad range of antibody services for individual or bulk orders. So you may ask, between polyclonal and monoclonal, how do we choose? So the choice of polyclonal and monoclonal antibodies is highly dependent on the applications. Being able to recognize multiple epitopes with high tolerance for differences in antigen and are easier to produce, polyclonal antibodies are often more for novel targets or time-sensitive projects. In fact, we at GenScript have polyclonal packages that allow delivery of antibodies in as fast as 45 days. Monoclonal antibodies, on the other hand, are the preferred alternative for well-established targets or when antibodies are required to recognize a protein with specific post-translational modifications, PTM, such as phospho-specific antibody, or when scientists require long-term reproducibility for high-stake projects such as in vitro diagnostic tools or vaccine development or drug development. So monoclonal antibodies become a safer choice as there is a renewable with batch-to-batch consistency, less background, and they are also highly specific. However, with all these advantages, it comes with a slightly higher price to produce, and also sometimes when it is too specific, it is less likely to recognize from a range of species. So to enhance the possibility of success for costly projects, sometimes costly projects such as monoclonal antibody production, and also to give the best value for monoclonal antibodies, GenScript uses time and tested technologies such as the hybridoma technology, and also use new innovative technologies such as the B-cell cloning and also the single B-cell cloning, which enable us not to only to accelerate B-cell selection, but also ensure we are able to capture a large B-cell repertoire. So to elaborate on the hybridoma technology that we use, upon harnessing the animal's immune system to generate antibodies of high specificity and affinity due to their intrinsic affinity maturation capabilities, we use the reliable and time-tested hybridoma technology to immortalize antibody-producing B cells using hybridoma cell lines to generate high-quality monoclonal antibodies. And just by growing them, hybridoma cell lines provide an unlimited supply of antibodies. And as a global leader in custom monoclonal antibody generation, we are constantly pushing ourselves to provide better quality and services using advanced technology. GenScript mono-wrap technology is able to generate a large number of high-quality rabbit monoclonal antibodies to maximize the success of identifying monoclonals perfect for your specific research application, especially for difficult applications such as immunohistochemistry, therapeutics, in vivo diagnostics, and in vitro diagnostics. And due to the rabbit's superior immune system, they can generate antibodies of higher affinity, specificity, sensitivity, and stability than those generated in other animal species. Animals with their robust and unique immune system also enables superior diversity antibody generation that is up to 10 times to 100 times higher affinity compared to rodents. So in addition, we have also upgraded our rabbit monoclonal generation platform to use B-cell cloning approach instead of the hybridoma technology for antibody discovery. So a typical workflow involves the isolation and enrichment and culturing of antigen-positive B cells from rabbit peripheral blood mononuclear cells, PBMCs, for screening and selection. So the desired monoclonal antibody clones are sequenced and recombinantly expressed. And through B-cell cloning, you can achieve rabbit monoclonal antibodies ideal for your research application in as fast as 13 weeks. And many benefits of this approach include that we can get PBMC only. We only get PBMC only, so that animals will not be needed are not sacrificed, and they are FACS-free to allow for high viability of cells. They are fusion-free, so they can avoid the B-cell loss, and this gives us an ability to obtain a larger cell pool of up to 10,000 memory B cells. However, despite the various advanced technologies we have, sometimes researchers still require much more clones to prevent missed opportunities and even higher success rates and time-efficient antibody discovery. So we can do that too with our Beacon platform. So it is the solution for customers looking for high diversity antibodies and are willing to invest for high quality antibodies with good diversity, specificity, and affinity. Harnessing the latest optofluidics technology, Beacon combines the isolation and screening and evaluation of B cells on a chip with the integration of high throughput platform for assay screening. GenScript has upgraded our lab generation capabilities by introducing a B-cell sorting platform with a variety of in-chip screening assays available to identify positive antibody clones such as like that. In-chip assays include antigen bit specific assay, multiplex IgG capture assay, and also reporter cell assay. Using the Beacon system from Berkeley Lights, our GenScript could screen using multiple binding and functional assays to select only the most qualified antibody leads in 24 hours. So this shortens the screening processes compared to around 12 to 14 weeks, approximately 3 months, using the hybridoma technology, or within 2 to 3 weeks using the upgraded monoRep. The hybridoma technology can assess only less than 1% of the B-cell repertoire, but the B-cell and cloning and the Beacon system can both fully assess the full antibody repertoire. This means that the antibodies discovered using the later two platforms are likely to recognize a wider range of epitopes on the target antigen, increasing the chances of identifying rare antibodies for challenging targets that may fail the hybridoma pathway. Having B-cell cloning and Beacon technologies enable us not only to accelerate antibody discovery, but also to capture a bigger B-cell repertoire and thus ensure a better antibody diversity and a higher clone number. So B-cell cloning can lead to more positive clones when compared to hybridoma technology. And this is especially crucial for challenging projects. Our data from 9 challenging projects, B-cell cloning yielded a higher positive rate and clone number than the hybridoma technology for the same project. as shown here. So based on our wide base of customer feedback, B-cell cloning has received the best feedback from our customers by having the high antibody clone number diversity and also a rather fast turnaround and a high success rate with challenging projects. Yet, they are most importantly at a cost-efficient price. So with all these technologies and guarantees, GenScript has served a broad range of customers with good track record. We fulfill researchers' investigation on novel drug targets for drug and biomarker discovery, food and agriculture, industrial applications, and also in vitro diagnostics development. All these applications rely on the high-quality antibodies to characterize, identify, and quantify novel protein or enzyme targets. So our antibodies can detect the target reliably with high specificity and affinity in a variety of research and developmental projects, including but not limited to immunofluorescence, Western blot, immunohistochemistry, ELISA, FACS, blocking assay, immunoprecipitation, positive and negative cell selection, and in vivo functional assays. So here we showcase a few case studies for our custom antibody generation platforms. So Kolosov and all had used previously our Poly-Express services to generate a polyclonal antibodies for a novel protein occluding in larval C. lamprey tissues. The polyclonal antibodies were used in Western blot and also in immunocytochemistry for novel protein characterization, and we help the antibodies help to define occluding localization and its abundance in larval C. lamprey tissues. Next, we show a research application in an article published in Science paper. The researchers, Han and all, had used our custom polyclonal and monoclonal antibodies in immunocytohistochemical studies and also Western blot to demonstrate the localization and the role of controlling GABA receptors abundance in the synapse in the central nervous system. The antibodies were very specific with low background, enabling the identification of SHISA7 in hard-to-define locales at the postsynaptic densities. The research also has significant physiological and clinical relevance for central nervous system mechanism of action understanding. Next, this Nature paper was published by the Vaccine Research Center in collaboration with GenScript. The research was trying to develop a HIV vaccine via the HIV-1 fusion peptide and investigating the potential of using the neutralizing antibody against this fusion peptide as a therapeutic strategy. One monoclonal antibody generated as part of the project was VFP 1.01 and was characterized in crystallography experiments to interrogate the structure and the function of this antibody. Alanine scanning is used to determine the residues necessary for antibody function, and surface plasmon resonance to determine the antibody binding strength. And two outcomes from this project were the antigen. We were able to show that the antigen prototype can be a good vaccine strategy, and two, the neutralizing antibodies that was developed can be developed into as a therapeutic candidate for HIV diseases. So here again, another research application showing our rodent monoclonal antibodies to detect survivin as a biomarker and enable monoclonal anti-survivin antibody to develop into a therapeutic candidate for tumor control and improve animal survival outcome. And in this study conducted by GenScript, monorep was used to develop against a very small compound, the ribtapanin, in animal feed. We use ribdotamin coupled to a human serum albumin as an immunogen, and the antibody developed using the monorab is specific to ribdotamin, while showing little cross-reactivity with similar compounds such as the salbutamol and the clenbuterol. Here, I would also like to introduce a great example of Scripps' commitment in joining scientists in the fight against COVID-19. Scripps had leveraged the unique advantages of hybridoma technology, monorab B-cell cloning technology, and the Beacon platform to generate high-affinity, high-diversity antibody against antigens, including the spike protein receptor binding domain, as shown here in this example. So spike protein has been recognized as a popular vaccine target to develop COVID-19 vaccine. Many therapeutics are also leveraging the functional neutralizing antibody that can block RBD ACE2 interaction, thus reducing the virus infectivity. So how do we go about the process of generating the spike RBD rabbit monoclonal antibodies using the Beacon system? After the rabbit immunization, we generate the single-cell suspension from one rabbit's spleen and subsequently concentrated antibody secreting cells using antigen and IgG positive B-cell enrichment process. Altogether, we imported over 15,000 cells onto the Beacon system and subjected these cells to antigen-positive screening using the antigen beads conjugated with RBD, and eventually we identify a total of 142 RBD positive hits. And in a parallel comparison experiment, we also did a single B-cell suspension to compare the hybridoma process with the Beacon system. So not only the monorab Beacon was able to introduce high affinity antibodies, we also achieved high diversity. This experiment compared the rabbit Beacon and the hybridoma platform, and as you can see, a total of 8 monoclonal antibodies were obtained in the Beacon experiment compared to 3 from the hybridoma approach. The 8 monoclonal antibodies from the Beacon system also had a high diversity to span across 4 epitope beams, while all the hybridoma antibody that was found was on the same epitope beam. The high antibody diversity could successfully enable multiple downstream applications, making them excellent candidates for in vitro diagnostic tools or other assay developments or potential further developments into therapeutics, thanks to having many candidates with a high neutralization efficacy. We evaluated the neutralization ability by evaluating the reduction of luciferase expression within the ACE2 stable cell lines that we have in Scripps. And listed here in the bottom right panel, you can see our top 4 rabbit monoclonal antibodies with superb neutralizing potency. Many of these antibodies has been very well received by the research industry communities and for various applications, example in assay development or even therapeutic antibody licensing. But not only so, we also compare it with the B-cell cloning approach to the Beacon system. And we were able to have a even higher diversity compared to the Beacon. A total of 60 monoclonal antibodies were obtained using the B-cell cloning approach compared to the 8 in the Beacon and 3 from the hybridoma. The 16 monoclonal antibodies from the B-cell cloning approach also had a high diversity to span across 11 epitope beams, and the high antibody diversity could successfully enable multiple downstream applications. And as many of you may be interested in the therapeutic antibodies generation, here we show our track record in delivering high-quality anti-idiotype antibodies for pharmacokinetic studies and immunogenicity assays. Anti-ID antibodies are powerful tools in pharmacokinetic studies to measure the drug level in patient samples. And the anti-ID bridging format highlighted in green box here is extremely popular for high specific antibody drug detection, especially when these antibody drugs can be of a very low concentration. So anti-ID antibodies can also serve as a positive control or reference standard in immunogenicity assays to identify anti-drug antibody characteristics potentially to explain the unintended side effects of an antibody drug. So given the important roles of anti-ID antibodies, sometimes scientists may encounter various challenges in anti-ID antibody production. The CDR region of an antibody drug may not be, for example, very immunogenic, and thus a little amount of antigen specific to the anti-ID antibody can be developed. And even if there are antibodies elicited, majority of the antibodies may be targeting the FC region here, and with less hits against the CDR. So these are some of the challenges that scientists face. And scientists must spend time and resources to optimize the clone selection from multiple, numerous anti-ID antibodies. And as mentioned, anti-ID bridging ELISA is extremely popular for high specific antibody drug concentration detection in patient samples. Researchers love the high specificity as an advantage of using anti-ID bridging ELISA format. However, oftentimes it is challenging to obtain successful anti-ID pairs. So thanks to the many years of expertise, GenScript has developed the domain expertise in developing anti-ID pairs, some which ELISA was developed to performed to select anti-ID pairs that could largely increase the success rate of immunoassay development. And as shown here on the right panel, monoclonal antibody 2 is able to pair with 4 other clones, monoclonal 1, 3, 4, and 5 for anti-ID bridging ELISA development. GenScripts also have a good track record in developing anti-idiotype monoclonal antibodies against CAR-T. The CAR-STFE region of the CAR is sequenced to generate a recombinant STFE. The recombinant STFE is then used as an immunogen in GenScript monorep services to generate a mature anti-idiotype monoclonal antibodies against the original CAR. We are proud to have the recognized track record to achieve high quality antibody against CAR-T, and also end-to-end one-stop service for antibody generation to immunoassay development. So last but not least, I would like to add a few antibody-related specialized services to further fuel your downstream development, research development needs. As we just mentioned, GenScript is proud to enable end-to-end solution for antibody generation to immunoassay development for a broad range of antibody projects. We provide custom ELISA kits development for standard kits and specialized kits. In addition, GenScript can also offer antibody conjugation and labeling using multiple technologies. We can label biotin, HRP, and many other fluorophore options. We also offer many additional services such as antibody purification, scale-up antibody production that I'll be covering in the coming slides. So after you identify an antibody lead using our platforms, one can improve its physical or functional characteristics and properties. And thanks to synthetic biology, scientists can engineer a library of different mutations and screen them to isolate the best antibody candidate. GenScript has established a pipeline for antibody engineering to further optimize the antibody lead quality to improve specificity, stability, solubility, affinity, or even remove hotspot degradation. The protein engineering uses the same principle to optimize the enzyme function, thermal stability, and also allow better folding of a new formatted fusion proteins. The antibody engineering capability can further improve our antibody lead quality, not only relying on the animal natural affinity maturation, but also provide a systematic approach to holistically evaluate opportunities to optimize antibody function. So in GenScript, we have a precision mutant library, which is a powerful tool to make antibody engineering possible. Like in this study, the objective is to establish a saturation mutagenesis library for 63 sites to all 20 amino acids. And from our next generation sequencing results, it shows that the ratio of the amino acids at each site showing here in panel 2, we observe equal distribution of amino acids across all the sites. And there is no stop codon introduced. So the precision control of the content has a lot of advantages, which then help us to minimize the downstream screening burden. And with a total of 63 to the power of 20, 1,200 candidates to screen is no easy job. So here, GenScript introduced our proprietary platform, fast screening of expression, biophysical properties, and affinity platform, the FASIBA, to enable high throughput screening for antibodies with the best antigen binding affinity, high expression level, and thermal stability. And as you can see, the top 5 candidates antibody was shown to have superior binding strength using SPR compared to the wild type. So now that you have a good antibody candidate, you may want to express it in large quantity for cellular or in vivo evaluation. Our high-density expression system allows us to increase the yield of transient mammalian expression by 2 to 6-fold using our GenScript's proprietary cocktail solution. These cocktails can boost cell growth and regulate the cell cycle and thus achieve a higher cell viability, which is essential for high antibody yield. So here we showcase a gram-level recombinant antibody expression using this transient expression system HD293F cells. On the left is the SDS page, and this is the Western blot QC results. On the right panel is you will find the purity as characterized by the size exclusion chromatography, demonstrating we are able to obtain 99% purity. So with all the information put together, I hope that our end-to-end antibody pipeline can best fit your research needs and help you achieve the research goal without going through all the hurdles one can encounter during antibody discovery for therapeutics, diagnostics, and research applications. And with this, I thank you for your attention. Any questions?
Hi. Hello.
Hi.
Yeah, all right. Thank you so much, Dr. Leong, for your wonderful presentations. Now we will proceed with the Q&A. So we will start to answer some questions from the floor. Okay, the first question is, how successfully achieve antibody specific for protein with few amino acid differences? For example, TNF, alpha, and beta free and bound form. What is the level of specificity which can achieve to quantify efficiently?
Okay, so we are able to, we have, as mentioned, we have the peptide algorithm, antigen algorithm for us to actually generate peptide. So we can actually, prior to generating the antibody for you, we usually have an evaluation to best give you the best proposal in how the generation of antibodies can be performed. So for such cases, like if we were to look for isoform specific, usually we will look at the sequence and identify sequences whereby, you know, they are unique to itself. And then using our algorithm, we will be able to generate the the peptide, choose the peptide sequence that will give a higher chance of antibody success. So in GenScript, we have the proprietary technologies to help you with isoform differentiation, so-called. Yeah.
All right. So next questions. Does GenScript provide training for antibody library synthesis? Does GenScript has antibody produced from fudge display technology?
Yes, we have the phage display system too. Yes.
Then how about the training for antibody library synthesis?
Training, in terms of training, yes, we'll be able to provide with technical support in the event. So yeah, just feel free to connect with us. So we'll give you the technical support that you need. My email is also attached here. So if you are interested to know more, yeah, feel free to contact us.
All right. So who will own the antibody produced through GenScript service?
Okay, so because GenScript is a CRO company, so it's a fee-for-service platform, so you will own the antibody.
All right. The next question is, what does Beacon platform do in antibody productions?
So the Beacon platform is just an optofluidics platform that we have. Basically, it is rather similar to the monoRAP technologies that have introduced. They both use the B-cell cloning technology. However, on the Beacon, we are able to use the optofluidics technology to have one clone to have one clone in one chip. So to have one clone, to test one clone at a time. So this allow the speedy discovery. May I show this? Maybe I could show this to you so that you can have a better idea on what I'm trying to refer to. Okay, here. So for example, so maybe this will be a better slide. The shown here is also based on the monoRAP technology. We'll harvest the B cells, and then after that, yeah, just hold on. I will show this one. Okay, wait. Okay. Okay, here. Okay. Okay. So each B cell will go through using the optofluidics technology, go into each well. So we can actually screen all these clones at one time. So we are able to have look at least 15,000 single B cell on the Beacon in 2 hours time. So different from the screening that we have using the hybridoma technology, whereby we have to culture and then we have to do the single cell, and after we have to culture again. It actually shortened the time for screening. So, but basically everything else is the same. Yeah. So it's just a very high throughput. It's for researchers who are, keen on having a lot of antibody leads, and they want it to have the results, in a very quick turnaround time. And we are able, using both the monoRAP and the Beacon, to harvest the full cell repertoire because of this. Yeah.
Okay. So the next question is, there's a very clean polyclonal antibody staining in ICC results. How do you compare GenScript polyclonal antibody with commercial and polyclonal antibody bought from the company?
Yeah, because we do a lot of QC, we guarantee the ELISA 1:128,000. In addition, we also provide QC, additional QC if you would like. So that's why we are able to generate, antibodies of good quality. And especially if you are to go for the monoclonal packages services that we have, we are also very flexible in terms of the services that we have, we are able to midway through the generation, allow the customers to actually test on their applications before proceeding on to generating the hybridoma clones that we would like. So we can even test on your application. Yeah. So that's why we can produce the quality that you require.
Thanks. So that question is, what does gram level means on slide 40?
The gram level means the amount of antibody that we are able to produce.
Okay. So how do you counter the challenge of non-immunogenic target?
Oh, we have the ImmunoPlus technology. And also we have our proprietary adjuvant technology to actually boost such hard to induce targets. Yeah. And
Okay.
I will actually recommend the monorack for these instances because of the rabbit's superior immune system for such targets. Yeah.
Great. So what is the amount of monoclonal antibody produced is typically generated for each project?
Yes, it typically for each project, but around 50 mg. Yes, but it depends on the project. Yes.
All right. So the next question is, what is the turnaround time for the monoclonal antibody generations?
Turnaround time, we have the depending on the host and depending on the the packages that we have. For the monorack, we can generate in as fast as 13 weeks.
Okay. So what are the QC methods available for the antibody generated?
QC method typically is ELISA, but we have Western blot, yeah, and also IP for some packages.
Okay. And why does rabbit have a superior diversity of antibody generations compared to rodents?
Oh, this is because of its innate system. It's actually mature. It have several times of somatic hypermutation, and also it uses a different system, the gut system, that is more similar actually to the human in this sense. So because of the gut system and also because it has several somatic hypermutation, so it allows a higher diversity. So in a sense, it's superior, and also because it is it is more heterogeneous compared to the human. It is further away from the human, so that's why it is you are able to generate higher diversity on human antigens.
All right. So we come to the very last questions from the attendees. What is the accuracy of beacon antibody sequence leads?
What's the accuracy of the antibodies? Because we do not have the benchmark, like the control for the antibody itself, right? Because it's for different cases different. So, but our antibody sequencing are accurate, are very reliable.
Okay, so thank you once again, Dr. Leong, for your great presentations. And we come to the end of the webinar. Thanks all of you who joined this webinar today. We hope you find this webinar useful and beneficial for your work. So thanks everyone, and we will see you again in the next webinar. Have a nice day and goodbye.
Okay, thank you. Bye-bye.